and good morning. So, I'm going to close the door. I get self-conscious talking to myself. All right, so this is Molecular Genetics. I'm your friendly neighborhood gene cloner, Dr. Galuski. So, we're going to go ahead and call the roll. When I call your name, raise your hand, answer here. No, I'm not going to call the roll. All right. So, I posted the syllabus to the Moodles. We'll get started on Tuesday. What I'm going to try and do is develop lectures for Tuesday, Thursday. And as we get a bit further into it, we're going to figure out how we're going to do the labs. One of the things is that we can do virtual PCR. We're going to be doing a fair amount of genome analysis. The U of I has their entire biotech genome center organization, and that we actually did a presentation on. They did a presentation back in Molecular Cell, and they do their whole thing these days is online. So there's a fair amount of molecular genetics we can do in terms of cloning strategies and learning how to do the basic techniques of recombinant DNA and biotechnology. Certainly the current environment provides us with a number of interesting things to talk about. Viruses. We'll be doing some good discussion of virology. Also, vaccines. So, one of the things you can do to make a vaccine is you can have a heat-killed virus. That's what the original, smallpox and polio viruses were, or an attenuated virus, a virus that is mutated such that it's not particularly infectious, but it does give you the immunological response. We'll be talking about the immune system as well. The modern way of making vaccines is to take a portion of the virus and express that in probably a bacterium, purify it from the bacteria, and use that as the immunizing agent. One of the brand new things that has actually been worked on is actually injecting the RNA to the virus. The RNA gets taken up by enough cells, they will go ahead and translate that RNA, produce protein that's foreign. It gets actually dumped out probably or presented somewhere on the cell surface, and that will also begin to elicit an immune response. We're kind of far away from having a vaccine, I'll be honest with you. But one thing I'm actually hopeful about is that Abbott Labs, our dear friends up in the North Shore of Chicago, they just announced yesterday the full rollout, and they got the FDA emergency approval for an antigen-based SNAP test. That is the term they use. It's what they use in the doctor's office when they're checking you for strep throat, and they'll swab the back of your throat, and then they'll say, okay, wait a few moments, and you sit there, and before you even leave, they'll come back and say, okay, yes, you have strep throat, here's your antibiotic, or no, you don't have strep throat, you suck, go home. It is very much like a pregnancy test as well, in which you've got an antibody affixed to a little strip, and you take the swab, which has the virus, and the antibody will bind to the virus, and if it binds, you get a little signal, and if it doesn't bind, then you don't get the signal. And it is pretty cheap. They're saying they're going to market it for $5 a pop. They can make millions of them. It's really fast. That's the number one thing. This issue with having to do, days waiting completely defeats the purpose of it, because if you are positive and you're still out in the population, you're going to be spreading it around. If you know you're positive, you need to get put into quarantine. And so this could begin, as it starts to roll out, change things. I was hoping we were going to have this kind of test sometime in the summer, and that when the fall started up, we'd actually be able to go through and test everybody for every 2 days or so. If you do that, you really can get yourself back to where you have a very low viral load. We ain't there yet, though, folks, and I'm not convinced that this is the only way we're going to be presenting class this semester. So ISU's got over 300 cases. We'll see how we do. I have my coffee. Oh, mother's milk. So, for those of you that don't know me, I'm Dr. Galuski, and I am the department's molecular and cell biologist, molecular geneticist. My PhD is in biology from Texas A&M University, primarily trained as a fruit fly developmental embryologist. Then from there began purifying proteins involved in metamorphosis, began cloning genes, and then my postdoc was at the MD Anderson Cancer Center, where I was doing a very large-scale genetic screen with flies and lots of cloning, and began doing PCR work there. Let's see. So here I do a lot of different things. In terms of a long-term project, we were working on the IL-1 cytokine and its receptor in hermit crabs with Dr. Handler as part of a chronic pain study. I haven't really messed with that in quite a while, though. Mostly what we've been doing is using novel techniques of recombinant DNA to begin looking at different kinds of questions that just come to my head. So last year we actually were doing a CRISPR-Cas9 experiment that was actually very simple, and I've been trying to think about other ways of modifying that. Currently in the lab, I've been working on taking a piece of the Zika virus and putting that into what's known as an expression vector. This is a DNA system that allows for bacteria to produce very large amounts of a given protein. And it's actually the way that insulin is made currently. It's been a difficult piece of DNA to clone for a variety of reasons that as we start getting into the technology, I hope to be able to explain it to you and so you can appreciate some of the difficulties. And it's one of these things that if someone were to give me a few $1,000s, I could probably have the clone for them in a week. But trying to do it a bit more old-fashioned, it takes a little bit longer. That said, we have a number of PCR machines here and a lot of electrophoresis equipment. Then we can run and carry out a lot of different technologically relevant experiments and things. We don't have a DNA sequencer, but we do send DNA out for sequencing. We may end up actually doing some virtual sequence analysis and things from samples that we have prepared or samples that we will prepare. Let's see. Another project I'm actually starting to work on and kind of thinking about is looking at the genetics of vitamin C biosynthesis in birds, particularly, just because Dr. Wilcoxon's got a lot of bird blood that I can mess around with. Just as a little side note, vitamin C biosynthesis is really a cool idea in that it is a single gene that is responsible for taking a substrate that is in a pathway that everybody's got and putting it into vitamin C. And it is a biosynthetic process. It's a single enzyme. It does generate a small amount of oxidative stress, and it does use some ATP. But evolutionarily, it's an interesting question as to why we don't all have the ability to biosynthesize vitamin C. What you may not know is we do not have the ability to biosynthesize vitamin C, but different organisms do. Mice, for instance, do. Dogs can. They can't get scurvy. It has some pretty remarkable effects in terms of like wound response and maintaining a strong level of antioxidants that you can't get from just an oral taking of the vitamin C. Of course, if you don't orally take in vitamin C, you can end up messing yourself up pretty good. Scurvy is what happens, and partly the collagen fibers in your cytoskeleton or in your extracellular matrix begin to unwind, and you start to have muscle loss, bruising, and your teeth start to fall out. It's not a good thing. In this day and age, it's pretty hard to get scurvy, but I guess if you decide you want to do it as a class project, go for it. So I have some ideas that, again, as we start talking about the technology, I'll tell you some of my thoughts about how we're going to be trying to amplify up and sequence some of the or the enzyme, the gene for the enzyme, in some of these birds. So let's see, what else is there? So you look at the syllabus, we're going to cover some topics, lots of topics. There are a couple of things that I want to make sure we hit on. Some of this is sort of a review from genetics, a review from molecular and cell. Other stuff will be completely new. And for sure, there's things that you're not going to get in any other class, like, unless we're teaching immunology, the mechanism of the immune system. We don't really have a virology class, so we're going to be talking about viruses and viral replication and different kinds of viruses. We're going to be talking about different kinds of gene regulatory systems. And one of the things we often do is sort of look at the prokaryotic models and how they're regulating genes, and then look at the eukaryotic models and how they regulate genes. And that's generally how we kind of finish up the class, is looking at some of the really important things in gene regulation in advanced eukaryotes, such as yourself or your dog. So another part of this class I think is kind of interesting, in that molecular genetics is not just a discipline, it's also a full technology. And the lab is where we explore some of these technologies, but the truth is a lot of what is going on in modern molecular genetics is outside the purview of most undergraduate labs. And so, we'll be talking about microarrays and different kinds of genetic analyses, all kinds of next-gen sequencing systems that most universities don't have, unless you're at U of I and a graduate student. You never even get into these things. So that's part of it as well, is that learning the technology and learning the vocabulary of the technology is an important part to understanding how you design a molecular genetics experiment, what kind of things molecular biology and biotechnology can do for you. What can it do for you? Just as an example, when Dr. Wilcoxon is doing some of his analyses on the viral load in different birds, they use an enzyme-linked immunosorbent assay, or an ELISA assay, the standard antibody test. And one of the reagents required for that is the piece of the viral protein, or the piece of the virus that you're trying to identify. And you want to see, we did this with West Nile, and we were trying to do this with Zika. So he tests birds for West Nile virus routinely, and I actually think that some of the birds that are migrating from South America might very well be carrying Zika, and it'd be interesting to know. So what you can do is coat a plate with the protein from what you're trying to detect, so one of the viral coat proteins, and then you take serum from the bird, put it in there, and see if it has antibodies that bind to it. We'll get into this. The point is, production of that protein is a little expensive, and so he had a supplier that was providing him with a portion of the West Nile virus, and I began looking at it and thinking, I might actually be able to make that for you. So it turns out there's a company, I think out of China, that actually sells a lot of different viral genomes. And they sell everything from Ebola, and I would imagine they probably have COVID. One of the points is that they always sell it either in a DNA form, and it's an RNA virus, or they don't provide you with the intact, complete virus, so you can't bioterrorize and build your own nasty superviruses, at least not with their DNA. But what they will do is give you a sufficient DNA sequence that will carry the envelope protein, the spike protein, those genes. And what I'd then do is amplify the portions I want up using PCR, clone them, and put them into these different expression factors. So with West Nile, we did the first run of it, and we made a grand total of enough protein for 2 or 3 individual wells, not a whole lot. So the expression system I was using was not one that was designed to produce large amounts of protein. I then shifted, and we've been using, for the past 9 months, we started I guess in January, working on a new system that generates very large amounts of protein. It's just a little bit harder to clone into it. So those are some of the things that we've been doing. Another thing that I've been interested in as well is using DNA-based systems for identifying species. And we started doing this a while ago using something that's called the barcode system, which is sort of a trivial term for the idea that you can, as it turns out, there's a mitochondrial piece that is able to serve as a species-specific identifier. And you have to pardon me, all of a sudden as I'm doing this, I'm thinking, oh my gosh, I hope the microphone's on, because I have the volume on mute so that all the dings that come through don't pick up. If it's not on, I may send it to you with subtitles or something. But regardless, so barcoding is a way of amplifying up a sequence that is specific to individual species. And we've used this to analyze bacteria. We do it with all kinds of different insects, all sorts of things. From there, that led me into some interest in forensics, and I am actually a co-teacher in our forensics class, which is a non-majors class, and learning forensic technology, is beyond what the criminal justice program is trying to do. We're probably mainly giving them just sort of the an exposure to the technologies. But from there, I've been interested in doing human identification. And that's led me to starting to work with some of the repeat sequences that are part of the Combined DNA Identification System, also known as CODIS. So if you ever watch Bones or NCI or whatever sort of forensic show you're into, you may hear about doing CODIS searches. And this semester, Lucy Koger and I are working on a couple of different CODIS sites to see if we can't resolve them. Partly, they typically are resolved using capillary electrophoresis, but I think we can do it using polyacrylamide gel electrophoresis. So that's me. We'll start out on Tuesday getting into some of the history of molecular genetics and talking a little bit about the notion that genes are in fact DNA, some of the very basic discoveries, and fairly quickly start moving our way into the structure of the gene. And then we'll actually take a little intermediate break and kind of part lab and talk about the biotechnology, how we actually can identify genes and some of the techniques and things there. Part of this is, as we proceed, at least like exam 2 is going to be what I would call a take-home, but it's all take-home, whatever in the BAS it usually is. It's going to be a couple of papers that you'll be doing some reviews on. The final exam will also have some papers, because one of the important things about this biz is learning to read the current literature. And truthfully, in order to read the current literature, you kind of got to start out reading some of the older stuff 10, 20 years ago that is a bit simpler, that you can then begin to see where the field is going. So that's part of how we'll be carrying the class out. We got some exams. We'll see how they go. I mean, at this point, they're going to be just take-home type things. Take home? You already are home. Whatever. And as far as lab goes, I'm starting to work on developing some sequences, and I'll try to develop sort of an organization of where we can start and where we'll finish. But we're going to start out doing some cloning strategies, and there's actually a number of interesting software pieces that you can download for free that you can actually do some recombinant DNA work on. And also with NCBI Blast, you can do virtual PCR on some genomes, and I'm probably going to have you do some of the analysis for finding PCR primers for the chicken vitamin C gene and see if we can apply those to different animals. So I'll use you as cheap labor, free labor. No. All right. That's about all I have to say for today. The class is typically a rather loose discussion type of class where I have ideas and I'll be gassing on for a while, and someone will say, hey, what about this? And we can go on. Feel free to come by. Really? I get lonely. I'm the only guy up here on the third floor in this corner. We can talk over ideas. If you are interested in research ideas, we got them. Yeah. So, until we video again, this is Dr. G. I'm out. One last thing. If you can come up with a good sign-off, I don't know what it's going to be, let me know, because I'm trying to come up with my sign-off. I mean, I'm out. That's trite. That's the way it is. Typically in lecture, Molecular Cell, I often slap the book closed and say, that's all I know. I don't know. It's all a little artificial. Hey dudes, take it easy. Stay safe. I'll chat with you later.